And before we go through that, actually, I want to talk about a few different housekeeping things real fast. Number one, you may have noticed that I wasn't here a couple days, and we missed a lab, and we missed a class. And you may have also noticed that I've been getting out assessments less quickly at the end of the quarter than the beginning of the quarter. And so for those reasons, we skipped a lab. We have skipped at least one assessment that was due to the protein structure prediction section. And then also I've dropped a couple because they were a little bit repetitive, and since I was getting stuff up late anyways, I decided, hey, you guys don't need to calculate both the maximum likelihood and a Bayesian phylogenetic tree. Let's just give you a maximum likelihood tree and a few other things like that. So actually it turns out that we don't have a total of 50 points in the class like I first believed that we did, or that we would. We actually have 42, which means that the breaks are going to come down as well. You have fewer assignments to complete to get the particular grades. I just wanted to inform you of this. So essentially what it is the break for an A is now like 39.5 and 38 points for A minus, and 36.5 points for a B, and then on. So now I'm going to post this onto D2L right after this, and of course you can always come back and watch this video as well and stuff like that. So but just to let you know that that's going to be up there. Also, just to kind of let you know, I'm still working through the grades. I ended up with like some 200 some odd assignments in there to grade over the weekend, and so I worked valiantly on it. I did not get through all 200 over this weekend, so sorry about that, but I am going to push through and get all those done. So, yes, that's kind of where everything is standing with that so far. I will definitely by this weekend have everything graded, and you will know what if anything you need to poke around with. But yeah. Okay. So back to what we're talking about. Again, we started last week with, on Friday of last week, talking about data mining transcriptomic data. So up until that point, we have gone through everything from you have your sequences hot off the sequencer in your FASTQ files. How do you go from there to looking at things like differential gene expression and things like that? We went through that in lab, and then we went through, once you have all this data and all this data has now been made publicly available, how do we sift through that data? And we talked about one way of going through that, and that was the Gene Expression Omnibus. That is one of the databases of the NCBI, and now we're going to go through another one here, and that is the Cancer Genomics Data Server. And this is another database of just raw data, including transcriptomics, but many other things, of a lot of different cancer genomic studies. This is a data server that's hosted by the Memorial Sloan Kettering Center. It has something like 126 data sets. So this is a ton of data sets. It's not nearly as many as something like Geo has in it, but it's still a absolute ton. And this is data from 26,000 patients. And all that being said, this is getting larger all the time. I actually went back and looked what they had in the middle of last year. Is just barely over 100 data sets. So in the last year, they've added like another 20 data sets to here. I think it's gone up like another 5,000 patients have been added to this. So what this has is a database of just the raw data from the cancer genomics. This is now the access that you get to this, to the Cancer Genomics Data Server, is through this thing called the CBio portal. And this is where they make all of this data publicly available. Here's how you get access to it. And one nice thing that they've done with this is that they have implemented an API, which is essentially a way for a database to communicate to some other application that is outside that database, so that you can easily retrieve information. So for instance, one API that I've used in the past is when I went through gathering all that housing sales data, I had a lot of addresses, but I didn't have latitude and longitude with those. So Google Maps actually has an API that you can just go give them an address and they'll give you back a latitude, longitude. So anyways, that's kind of a long story, and I actually made them ban me for a little while. You make too many requests too fast and they like shut you down. you have to learn how to get around that. But nicely, with the Cancer Genomics Data Server, they actually made this, and they have implemented this API into a package in R that you can readily just access this data server and pull back information. So make requests of all that data and just pull it back really easily. They also have a web server, and they work kind of hand in hand, but the nice thing about the R interface is that you can manipulate the data in a lot of ways that you can't on the web server. There's a lot of constraints on how you can manipulate the data. And then also looking across studies is something that's a lot easier in R than the web server. They kind of make it a little bit hard. It's easy to look within a single study, but it's a lot harder to look across studies and collate the data from multiple studies. So the kind of data that's available for each one of these data sets, or most of the data sets, is first off, they have copy number variation for genes, which is kind of nice to have. And one thing that we're specifically going to look at and be interested in, they have the mRNA expression data as well. So they have this both from RNA-seq studies and then also microarray studies. Not necessarily both at the same time, but many of these studies actually have concurrently both the microarray data and the RNA-seq data for the exact same samples, which is kind of nice depending on what kind of data you have that you want to compare this to. It gives you some flexibility. Also, for in my browsing around, it looked like somewhere around 50% of them, they have micro RNA expression for these as well, which is another really nice thing. Mutation profiles, protein and phosphoprotein expression, DNA methylation expression in all of these, which is also something that isn't necessarily easy to get a hold of in other data sets. And then here's actually one really big thing. So when we're looking at Geo, all the data sets in there were very self-contained. You could go and you could access all of this mRNA data or all this expression data for like a bunch of mice or something else like that. And that's all well and good and that's great, but you don't have a lot of other corroborating data with that. You don't have a lot of other data that gives you a lot of idea about what else is going on with that. Now that's really great, and we could look at things, like for instance, we could look at, Tyler will enjoy it. Yeah, the Menton, and I'm getting the glare again. Did anyone notice that he was glaring at me the entire time last week? I'm sorry, Tyler. Anyways, but we can look at the expression of fermentin in 9L versus normal rat brain cells, and that was great, but really that data was kind of, everything else was kind of divorced from that. We just had like this expression data and we could put it into two groups, but that was about it. In this case, actually, we have tons for all of these individual patients that you have in there. You have tons of clinical data to go along with that. Of course, all this, due to HIPAA laws and stuff like that, the identifiers have been removed from this, so you can't like track down who they individually were. But you have things like survival past the time of diagnosis, sex, other things like this. I'll go through a little bit more of that as we go through this, but there's all of this other clinical data that's really valuable that you can go and you can compare then to the transcriptomic data or whatever other kind of data that you're pulling down from the server and do some analyses that way, which is really great. Because one of the things that we really kind of are interested in, like for instance, is survival rate with different kinds of cancer, or different kinds of expression in cancer, so tends to be kind of what we're interested in. All that being said, we don't necessarily have all of these data types for all of these data sets. It's kind of hit or miss with some of these. mRNA expression is there with absolutely every single version. Every single patient that I've ever seen in there has mRNA expression data. I do not think I've seen one that has not yet had both RNA-seq and microarray data. Not all of them have microarray or microRNA expression, excuse me. That's, 50-50 of them. The epigenomics DNA methylation stuff, that's there for about 50% of them. So those ones are kind of like, okay, hit or miss. But like mRNA expression, there for every single one. Okay, so then the obvious question here is then where are they getting all this data from? Now, the nice thing about this particular data server over something like GenBank or Geo that we talked about last week, all of those are essentially, not quite, I shouldn't quite say a free-for-all, but essentially everyone that's getting this kind of data is uploading that data to these servers. So if most people who have gone through and published in the United States have published transcriptomics studies, have then uploaded that transcriptomic data to Geo, and then there's an accession number with that, and really the quality and the type of data that you get there is all over the map. It might be good, but you have to really do your homework to make sure that that data is really kind of solid and is what you want. Now, the nice thing about the Cancer Genomics Data Server is that this is a curated database, which means that they are going through, often from places like Geo, other sites like that, and they are choosing studies that are high quality, have a high number of replicates, and then they include those into this data server. Once they do that, they're just not including them whole hog and just like kind of taking them as they are. They actually get them all into the same units. They're making sure they're normalized roughly the same way. They're making sure that the same values are calculated for each of them. And so this is really nice, which means that as you go through and you interrogate this data, you actually have to spend a lot less time you still need to do enough background research that you know that you can trust your data set. You are always, when you go out and you publish any sort of data, it's ultimately on you, the quality of data that you present to other people and publish. But that being said, you can have a lot more confidence in this data set than something that you just pulled down from Geo or something else like that. So the nice thing about this kind of data and a curated data set is that you can just have a lot more confidence in what kind of data is there. A couple of the big places that a lot of this has come from, number one is the Cancer Genome Atlas. I think roughly about half of all the data sets in here have come from the Cancer Genome Atlas, which was one of these large projects to try to, that included among other things, try to look at transcriptomics from a wide variety of different cancer types. So I have the Cancer Cell Line Encyclopedia. And the NCI, this is the National Cancer Institutes. They have a group of 60 different cell lines, cancer cell lines, that, again, these are kind of curated cell lines, and they've done transcriptomics on all these. And this is another group of data that's in here. And actually several others from several different large studies that have been done and other stuff like this. So there's other kinds of just, I wouldn't necessarily just say one-off, but other large individualized projects that don't quite reach the level of like a Cancer Genome Atlas or something like that. Okay, so let's take a look at what this looks like in using this in R. This is something that we're going to be doing in lab this week. And so I'm going to give like a quick little preview through this. I'm going to do this. So we'll go through this and kind of look at. Oh, by the way, hey, how much nicer does that look? I spent some time in here this morning fixing the projector so you can actually read it. It makes me very happy. Okay, so the package that we use for this is CGDS, so the Cancer Genome Dataset, and then R because we're using an R. And the basic workflow of this is going to kind of be the same all the way through. So the first thing is you have to set your the path to the actual public portal for the CBio portal. This is the same all the time. I actually don't know why in the world they decided to do it this way rather than just having this integrate in there, because the only site that this package works with is this one. And the funny thing is it's actually changed. Like the URL has changed in some of the early editions of this. And so, anyways. But one of the very first things that you can do is that you can and one of the first things that you need to do in your workflow through this is get all of the studies or all of the data sets that are associated with this, so you can kind of browse through them, see what kinds of things you want to be looking at. So the first thing is to grab your studies, and you do this with this command, the get cancer studies. So I'm going to write this up here. It's kind of the first step of what you need to do to actually start looking at data in this data set. So we do that. We can come back in here and look at what that looks like. And so all of these, what they have is they have three different things. They have a cancer study ID. And this is really what you need to grab out of this, because this is the name that you're going to use to grab data from each of these data sets from here on after. After this, they have the name of the type of cancer that has been done, and then also something of the reference of where this came from, and then also a description of what just kind of a short, brief description of what kind of things are in there. So, for instance, here we have acute myeloid leukemia. This is from TGCA, or the Cancer Genome Atlas, and it was published in the New England Journal of Medicine 2013. And then go on, we have bladder cancer, published in the European Journal of Urology, 2014, and then just on down. So we have all these data sets in here that we can get access to. So we can pull that in. Usually I pull this in as, let me close a handful of things up here. Pull this in as its own object in here, so then I can just, when I need to call those different studies later on, I can just grab the cell from there that has the individual thing that I want to look at. The next thing, I just did this here because I'm going to it was so much fun going through Vimenta and gliomas last time that I think I'm going to do that again, just to, don't be too irritated, please. It's not in humans this time. We're not curing gliomas in rats anymore. So you shouldn't be quite so irritated. So we have, what I'm going to do now is actually make a subset of these studies, and I'm using a regular expression. So one of the nice things that I haven't talked too much about in here is that you can use regular expressions that we talked about earlier in R to kind of get narrow down our data sets using this grep function. Actually, well, I don't want to talk about that too much right now. But grep, and then we can look at glio, so we can get anything that has glio in the name. So this will get gliosarcoma, glioblastoma, a glioblastoma multiforme, just a glioma. Anything that has glio in it is going to get captured in this search that I do right here. And I'm looking in this second column here of studies. So if we go back to studies again, I'm looking down this second column, which is name, and I'm coming down and looking for any glioma in here. So like here, brain lower grade glioma is going to get captured by this because it has glio now in there. So we run that, and that then is going to bring up a data set of so now it's essentially just that same data set as we had before, but it just has these gliomas. We have a brain lower grade glioma, we have a glioblastoma, another glioblastoma, then we have a glioblastoma lower form, and then these lower grade gliomas. So we have 5 different gliomas that we have captured with this. And then the next thing that you have to do is actually go and look at the data sets that are within that larger data set. So there's multiple ways actually that all this data can be divided up. And the people that have imported this into here have actually taken all these data sets. They have pre-divided them in different ways that you can look at, and we need to know which way is being divided and interrogate using those divisions. So this will actually make a little bit more sense if I actually just do this. So I'm actually going to grab that first one. This is going to call LGG for low-grade glioma, and then enter in get cases. So the command here is get case lists. And I just want to do this. Now, the 2 different things I give it is, again, this website, so it knows where it's talking to. And then I want gliomas, and we need to give it, again, the ID number, or the ID of that specific site. So if we go back to gliomas up here, this is what we really need to interrogate. This is this LGG_TCGA. This is what we need to tell it to give us this study. So we can do that by getting gliomas, and then row 1, column 1 gives us that. And then actually, again, telling you just to go there to grab that. So we can get that case list. And then if we go look at this, this is what we get. We have an ID now for each of these case lists. And what this does is this also gives us each way that this data can be divided. So we can have all complete tumors and all tumors. Essentially, this is all the data that is within there. All complete tumors gives you everything with all of the data associated with it. All tumors just gives you everything. And then we have sequence tumors. We just have everything that's been sequenced. This will cut out everything that we do not have sequences for. Everything, all tumor samples with copy number alterations. CNA is copy number alterations data. So we can just then subdivide this data by, okay, just give me back the data that has copy number alterations. One that we're going to be interested in, we come down here, tumor samples with mRNA data. And this is by RNA-seq. But if you come, actually this one does not have microarray data. Interesting. But it does have RNA-seq data. So we can, this is one that we're particularly interested in. Not all of the patients in here had RNA-seq done on the tumor that was associated with them. And so what we can do is we can then divide by calling this particular ID, we can call just that data that has RNA-seq data associated with it. So we come back here, and then the next thing we need to get is what we know as genetic profiles. And the genetic profiles now are the particular measurements that were made. So again here, this is going to look kind of similar to what we just saw, because that is the data divided by the different measurements that were made. Now this is the different measurements that were made. That probably sounds confusing at this moment, but just imagine this. Imagine what you wanted is you just want to look in the population of data that had RNA-seq data. But I wanted copy number alterations. I want to know how many times is there a deletion of this gene, a duplication of this gene. It's exactly the same as it normally is. But I want that for ones that also have the RNA-seq data, because I want to go on and later on compare that to the RNA-seq mRNA expression. So you use the case list for the mRNA data to actually pull down the copy number alteration. This will make more sense. For the moment, let that kind of seep in just for a moment. Hopefully by the end of class today, and then also at the end of lab, that will make more sense once you've heard it and seen it in action again when I do it, and then actually as we sit down in lab and do it ourselves. But I can understand that's a little bit confusing at the moment. But this is the different data that we have. So again, we have two ways of looking at the copy number alterations here. mRNA data. We have two different things here also for mRNA data. You have, number one, you have the actual data here. Now normally this is in what we've been talking about before. This is in the reads per kilobase per million. So it's in that normalized data that we've been talking about, or that normalized units that we've been talking about. But we also have a z-score here. Now the z-score is nice because this gives you something like a p-value that gives you the deviation, kind of a standardized deviation from the mean of the data, which is very nice as well. And we'll talk more about that. Okay, so once we have the genetic profiles and we have the case list, the next thing that we can do is then actually go and get the data that is associated with those different actual individual samples. And the command that's associated with this is get profile data. Once again, like with every single command that you have to do where you interrogate the server on this, you have to start out with this website again. So we put in the object that I put that website in, mycgds. The next thing that you give it is you want to look for the gene, or you can interrogate any specific gene that you're interested in. So we're looking at mRNA data is what I'm going to be looking at. So I need to give it a gene. Now I can give it any list of genes I want. If I really want to and like tie up the server for a while, I could tell it all genes, but I don't really want to do that in the middle of class right here. So what I'm going to do is I'm just going to look for Vimentin. You need to use the standardized gene code for these. So that for Vimentin is just VIM, and when you put that inside quotes. So here is the gene that, or genes that you want to look at. If you want to look at multiple genes, again, just use the C function to combine different ones again. And then the next two after this is we want to look at the genetic profile and then also the case list. So what specific data do I want? So let's go back in and looking at our genetic profiles again. And what I want to look at is mRNA expression here. So I'm just going to grab that. So that is, the ID for that is in column 1 and in row 6. So if I go back and look at this, once again, I have row 6, column 1. That is the data I want to pull down. And then again here, I got to look at what case list am I interested in. And in this case, I grabbed all tumors. Actually, that might be a little bit broad, because that doesn't necessarily mean all the tumors have RNA-seq data associated with them. If we come down here to tumor samples with mRNA data, RNA-seq V2, that's number 6 here as well. So actually, I'm going to change that to reflect that I want to get just that data that has RNA-seq data associated with it. And we go ahead and run that. And I'm calling this LGG, so for lower grade glioma.x for expression. We look at that. And this is what we get back. So again, we get on this, each of the row names is the identifier for the individual patient that was associated with the study. And then over here, the VIM. This is the expression level of Vimentin that was found by RNA-seq in that patient, in the tumor of that patient. And again, all of this is in the RPKM units. Okay. And then I'm going to add a column here, just type to that, so I get lower grade glioma, just so that I can somehow identify it after I combine this with other data. So that's what I get right there. Just added in what type of cancer that was. Okay, so essentially what I'm going to do is I'm just going to run this again, exactly the same workflow for 3 different things. This time I'm going to use glioblastoma that was published in 2013, their provisional glioblastoma data, and then also again here the acute myeloid leukemia, just as like a non-brain cancer to kind of compare things to. Run all those again. But again, I'm running the exact same thing. I get the case lists, and I find which data set I want to look at, or which way of dividing the data set I want to look at. I get the genetic profiles, and I look for which one has the mRNA. And then again, just do the same thing where I give it the website, give it my gene of interest, and then once again, now this is changing every time. Which actual row that data is in, you have to actually go back and look at them to find it. Actually, if you really wanted to run a fancy regular expression or something, you could find it. But the easiest way is just to go back and look at each of these data sets when you pull it down and find which one is the mRNA and which one is the case list that actually has that slice of data that you want. And then what we can do then, just some simple manipulation of the data that we've learned earlier, R-bind, actually take these data sets, put them together on top of each other, put this into one data set called expression. So here we have the lower grade glioma, the glioblastoma, the second glioblastoma, and the acute myeloid or leukemia. Put them together. And then we have, wait, nope. Give me the right one, please. So then we have this entire data set here that has Vimentin expression in each of our data sets as we go ahead and go down. So there's the brain lower grade glioma. You see we have hundreds of individuals for each of these. So there we're getting down to the end of the brain lower grade glioma. I think we have a couple hundred of those to look at. So we actually have a really nice robust data set. Glioblastoma down under this. That continues on. Okay. So let's head back to our data set. So now what we can do is we can analyze this any way that we want. We can look at what is the Vimentin expression in these different data sets. And here's the plot of that. So what I pulled down, actually the last one I pulled down was not glioblastoma, it was the glioblastoma multiforme. Is that what 9L is? No? Okay. So we have the acute myeloid leukemia, brain lower grade glioma is, oh, never mind. Glioblastoma actually looks like expression is elevated compared to these two, but without a control in here, it doesn't really tell you, is it depressed in this or is it elevated in this? And then also glioblastoma multiforme. This isn't any particular, well, it is particular because I like Vimentin now because of you. not being in the field, I actually don't specifically know if this is a useful comparison or not. The point here just is that actually this came from 4 different studies. This is now standardized normalized expression from 4 different studies. You would, if you were going to actually use this data, have to go back, look at those studies, make sure that you can, based on their methods, these are comparable things that you can actually compare to each other, other stuff like that. But once you have decide that it's really relatively easy to pull the complete expression data from 4 different studies that had tons and tons of different patients in it, and get some really interesting expression data here. We could also go through and we could start doing statistical tests on this. You could easily run an ANOVA on this data and move on from there. But I mean, again, just pulling one of these is 4 lines of code in R. So really, really a very easy way to go about that. Okay. Wait, what was you saying? Oh, there was one other thing I wanted to talk to you about in here. The other really nice thing, and again, the really nice thing that makes the Cancer Genomics Data Server really powerful compared to a lot of the other places that you can mine for data or look for data with expression stuff, is that you can actually get clinical data with this. So here we have get clinical data. And once again, you're just going to repeat what you had up here. What we have is we have a case list now. We don't have to give it a gene because the clinical data has no particular gene associated with it. It's associated with the patient, not with any particular gene. And we don't have to get any data set because it's not associated necessarily with the mRNA expression or the microRNA expression or anything else like that. It's associated again with the patient. So all we need for the get clinical data is, again, the website that we call, and then just the case list or the way that we want to subdivide this data is all we really need for that. So we run that. And says gbm.c. Okay, so here's the clinical data. So this is for the glioblastoma that we had. Might have been the glioblastoma multiforme. But you have, when the sample is taken, you have the age of the patient, the days after birth from the patient, the days before death of the patient, a little bit morbid. Last days to follow up. You have status, whether disease-free or regressed, the gender of the patient, and a lot of other things that informed consent verified. Hopefully that's yes on all of them. Karnofsky performance score, I actually have no idea what that is. The status, living or deceased, and then a bunch of other things. Performance status, timing, post-adjuvant therapy, preoperative. So you actually have a lot of data to go with this. And so this becomes really powerful because you can actually look at, like, for instance, since you have whether or not they're deceased or living, and then you also have time before death between this, you can also look at differential gene expression, look at survival curves of, okay, so is a different survival expected for different kinds of expression, things like this. And again, once again, this is the kind of information that is very, very, very rare with other transcriptomics databases that you can get access to. So for instance, if I go back, I just had like a little... I combine that with... So I can look at Vimentin expression and actually how long they survived after that. And... Oh, no, I need to add that, and now add that. Too large to fit the margins. Let's make it bigger then. Vimentin has nothing to do with how long they lived after that. Sorry, Tyler. Okay. So anyways, that's just a little kind of preview. We're going to be going more into specifically using that specific data server in the lab tomorrow, but I just wanted to give you a rough through of that. Now, so kind of summing up the idea of mined data is there's several different ways in which this can be used. One of them is that oftentimes people are using this to create hypotheses. You actually go look at this data, look at different associations, different interesting trends that you were seeing in this, and then use this to actually go out and design further experiments. Now, this is probably one of the most rigorous and best ways to use this, and the reason for that is something that we've talked a lot about before, is that when you have gobs and gobs and gobs of data, one of the biggest challenges that you run into is a false discovery rate. If you have lots of data, you are going to find interesting trends and correlations by chance alone. If I'm looking at thousands upon thousands upon thousands of data points, I will find something just by chance alone. And so one of the best ways and most rigorous ways that people tend to use this is to use this to look for, oh, hey, we can find all these potential interesting trends and associations, and then use that to design experiments to actually go seeing, are those real? Or is that something that is just kind of statistical fluctuations in the data? And again, this is kind of like what we were talking about with RNA-seq, that using RNA-seq is the first pass, but you are going to find things. You're looking at 200,000 genes. You will find some that have differential expression by chance alone. And then moving on from that and doing something like qPCR to follow up to actually see, okay, is there actually differential expression between two different tissues, two different developmental stages, whatever it is that you want to look at. Also, oftentimes this is used to complement another experimental data set. I've seen this happen a lot where, okay, so someone has a great data set, but they can use some sort of data mine to actually lead into that or actually complement what they already have. And another method here is just completely standalone studies that are only done with mined data by themselves. Now, this is something that you always have to view with a little bit of caution because, again, like what I was talking about with just the perpetual kind of threat of false discovery, but there are quite a few still studies that just solely rest on data that has been pulled from servers like this. I took about 5 minutes last night and grabbed 3 of them, just really fast, that had to do with actually the specific package that we're looking at. So this one right here, looking at some particular gene and in ovarian cancer. If you look through this here, they're talking about getting their data and they're using it from the Cancer Genomics data server using the R package CGDSR, again, exactly what we're looking at before. That's not all they use, actually. You come down here and they got a Vienna ovarian cancer data set, and this came from Geo, using GeoQuery, so what we were talking about on Friday. So 2 different data sets, and this is everything for their data acquisition. This is it. So their entire study in this case relied on data that came out of essentially what we've been going over for the past few classes. Again, a scoring system for predicting identification and prior tracing of predicted cancer genes. Again, completely out of essentially, well, no, they got 2 different data sets. The major one here is coming again using the CGDSR package that we're just talking about from the Cancer Genomic Data Server, and then another one we're getting somatic mutation data from another database. But again, the complete study was done with obtained data from these data servers. And then dysregulation of the X chromosome inactivation in high-grade ovarian serous adenocarcinoma. Again, in this case, almost completely. I think there's a couple different places that they're getting stuff from, but the majority of the data, so all the data is coming from the Cancer Genome Atlas, which you can get through the CGDS. However, they grab some of that through their web portal, and then the rest of it, including survival, tumor stage, grade, were all grabbed with CGDS. And again, this completely was done. And these are actually, like, the first one was actually in a journal that I've never heard of, but the next 2 were in PLOS One. They have good peer review. Not necessarily reviewing for how important they are, but yeah. So, I won't rant about PLOS One right now. So, okay, so that's just to kind of lay out that just mine data by itself, in some cases, constitutes all of the data people somewhat regularly, not as much as the other avenues I talked about, but somewhat regularly actually go through and just use that data. Okay, so I'm going to finish up now. I'm moving in. I want to talk a little bit more about a couple different things that are similar to what we've been talking about. And this is microbiome data and environmental DNA data. So just to kind of like, in case you've somehow gotten to this point in your biology career and had never heard of what a microbiome is, which I hope is not the case, is the complete collection of microorganisms that is associated with some particular environmental niche. And most often when you hear this referred to, that particular environmental niche is an organism, some other organism, like you. And oftentimes these are broken down into a particular region of that organism, say like, for instance, your gut microbiome, or your skin microbiome, or something else like that. And so that is essentially what we're looking at with the microbiome. Now, I want to go into this here. Well, and just to add in one thing here, we're kind of discovering a lot that these microbiomes play a huge role in an organism's physiology, and not just their physiology, but also their ecology. Your ability to survive in certain areas has a lot to do with the bacteria that's associated with you, and so environmental physiologists are paying a lot more attention to this as well. I point them out in particular because it's kind of what I do, so I care a lot about that. But in human health, they're beginning to be recognized as being vitally important as well. Now, that being said, I'm going to this next particularly, and I want you guys to kind of look at this because the same basic set of tools that you have sat down and you've used for RNA-seq data is going to come up again and again and again. That basic workflow that we've looked at, in which you go through alignment to or reference either data set or genome, you pull down the annotations for that or some sort of data that's associated with that. You go back and you do some sort of quantification and normalization of how many reads then that you have per feature, and then you do some sort of analysis based on that. That is something, that basic workflow is not something that is just specific to RNA-seq or to what we've been doing previously. That's something that comes up again and again and again, over and over in many times that you're analyzing high throughput sequencing data. It's not something that's just a one-off specifically for RNA-seq. So that, I want you to kind of pay attention to what we're doing there. I'm going to go through this relatively quick. We won't finish it today. But the point here is I want you to be kind of familiar about how do we go about analyzing this kind of data, and not just kind of in a vacuum, but how this relates to how we've already talked about analyzing this kind of, or similar data. Okay, so the first step in this usually, I'm skipping over the actual collection and isolation and extraction of DNA and going straight to the sequencing. But again, we're doing DNA sequencing by pyrosequencing, Illumina, or some. Now those have been the most common. But that being said, again, there's so many other high throughput methods of sequencing that are starting up or really kind of getting a foothold. And so really, almost any of these high throughput methods can give you the data that you need to go through microbiome data. And this can be in two different directions. We can do this two different ways. Number one, we can do this in a targeted fashion. And in targeted fashion, what I mean is that we're not just looking at like essentially trying to quantify all of the genetic material that's there, but we want to specifically look at some sort of sequence. And oftentimes this is something like 16S that has some sort of taxonomic significance. And so the way that we go about that is to first amplify by PCR 16S, and doing this with universal primers or something else like this that will essentially amplify all of the 16S from every organism or close to every organism that we have in that sample. And so now that we have more 16S than we have anything else, we can run that through sequencing, and the point of this really is to identify the taxonomic diversity that we have in that sample. Alternatively, we can just do shotgun sequencing where essentially, like we've talked about before for pyrosequencing or Illumina, similar to what we saw for RNA-seq, except without, not necessarily using the isolation of the mRNAs like we've talked about, but sometimes going with the entire, just the entire genome, going through and doing shotgun sequencing. So we're getting a sampling of everything there. All of the different genes that happen to be there. Now the point of this is not taxonomic diversity. This actually, you can get taxonomic diversity for this if you really want to get that. But you have to collect incredibly larger data sets to get taxonomic diversity from this. What you can get from this is physiological diversity, which honestly in some ways is much more interesting and much more useful for the study of how this works with the entire organism than just a big data set of what kinds or what clades of bacteria are in there. Okay, so very briefly talking about the targeted sequencing. Okay, so how will we go through processing this? The first thing is, so now we've gone through the sequencing, we've amplified 16S by PCR, we've gone through and we've sequenced that set, and now we have a FASTQ file, like what you saw in lab last week. And with that FASTQ file, we're going to take all of those fragments that we have in the FASTQ file, and again, remember, these are really short. These are somewhere between like 50 to maybe at really good times getting up to 200 base pairs, but we're talking like in the 100s, 50s to 100s. And we're going to do pairwise alignment. We're not aligning yet to some specific genome, some specific database. We're going to pairwise align between each of those fragments. So we actually get how those individual fragments line up to each other. And the next thing that we do is we go through and actually look at the genetic distance between these fragments. And so we can calculate this genetic distance any of the different ways that we've talked about before. It can be identity, it can be like the Chimera 80 or something else like that that we've talked about. We just get genetic distance between each of these fragments. And we go through and we do some sort of clustering operation that then binds together everything or groups together into a group, everything of a certain genetic distance. That is some cutoff that we have predefined. Now there's best practices for this, but we're going to take everything of some certain range and say, okay, all of this we are going to group together. And we're going to call that an operational taxonomic unit. We're not calling it a species, we're not calling it genus, we're not calling it family. There's a whole range of reasons why we don't do this, including the whole idea of a species in bacteria is kind of slippery as it is, considering that you can't put them together and see if they mate together, so the whole biological species concept goes out the window. But to use something called the phylogenetic diversity or the phylogenetic species concept or something else. But then also, this allows us to do something nice. Is normally now the really hard part is then taking all of these fragments, this is what takes a long time, and looking at them or aligning them to a database. What we can do now is take the consensus sequence for each of these operational taxonomic units. So we know all of these things are closely related. We know they fall within a certain range of a group. And we can go back and we can now compare just one sequence for each of those operational taxonomic units. This is going to cut down our processing time hugely. Because instead of going through and looking up hundreds, sometimes thousands of reads, we can collapse that down to one for this group. And this other one of thousands of reads down to one. So we can actually break down our computing time hugely. And also, we know already then, this does count the next step for us. So we're comparing this to this database. This is going to be somewhat similar to blasting it, but we're doing this slightly differently because the databases that we use again to cut down processing time are going to be smaller, only 16S, or whatever gene of interest we're looking at. And then we start classifying these to the lowest taxonomic group. And then now we know how many reads we had in each taxonomic group. We can do some basic statistics to try to estimate what proportion of our reads belong to each taxonomic group. So we have a group of taxonomies, or taxonomic groups that we can identify in that microbiome. We have a proportion of reads. So now what we have is a group, and we have a proportion of the entire sample that that group made up. Okay, I'm going to leave it there for now because we are running out of time. We're going to continue with this, finish up this, and then environmental DNA as well on Friday, and on tomorrow in lab, we are going to go through, actually mine the cancer genome data server. Okay, thank you very much.
Yeah.
And then pretty pictures.
Do you have a paper for me?
I can go down and print it off.
Yeah.
Yeah, okay. I'll go down and print it off from...
Yeah, I'm not sure if I have to go do it next.
Okay.
But if I'm not in my office, you can just put it in my mailbox.
Okay.
Right by the door.
Okay, awesome. Thank you. Yes.
Due to the fact that the wonderful little room, the lounge, is still under construction, it looks like we're going to be meeting in the library. I think it's called the.
I like the haircut, by the way.
Okay. I have no idea where that is. I will follow you over there.
Okay, yeah, I'll send out an email today
Okay.
also stating the actual name of the room. But
Okay.
Yep.
Sounds good.
Okay, thank you.
No problem. Okay. Sounds good. How's it going?
That is true.
Who are you?
Are you talking to me?
Yeah.
Okay.
I just asked you how it's going.
Oh, you're talking to me.
I didn't know.
I'm sorry, I was looking at, yeah.
I was wondering if I can meet with you sometime to ask you some.
Yes, can you send me an email
Yes, I
and
will.
what times that you're available, and then I will, yeah.
That'd be awesome. Because also, like, I was trying to do my Bayesian tree for, I think it was Lat 8, and it